Cellular respiration is the process by which glucose is converted into carbon dioxide and water, with energy being conserved in the form of ATP, adenosine triphosphate. What's important is that it doesn't happen in one explosive step. It's broken down into controlled stages so energy can be captured efficiently instead of being lost as heat. The process can be divided into four major stages: glycolysis, the link reaction, Krebs cycle, and oxidative phosphorylation. Glycolysis occurs in the cytoplasm and does not require oxygen. It begins with glucose, a 6-carbon molecule. The first thing the cell does is phosphorylate it using ATP. This traps the glucose inside the cell and makes it more reactive. A second phosphorylation occurs at the fructose 6-phosphate stage. This step, catalyzed by phosphofructokinase, is the main regulatory system of glycolysis. It's essentially the commitment step. Once this happens, the molecule will proceed through glycolysis. At this point, the 6-carbon sugar is broken down into two 3-carbon molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Only glyceraldehyde 3-phosphate continues directly, but DHAP is converted into it rapidly. So essentially we now have two identical molecules moving forward. In the energy payoff phase, each glyceraldehyde 3-phosphate is oxidized. NAD plus is reduced to NADH, meaning electrons are being captured for later ATP production. Substrate level phosphorylation occurs twice per molecule, once at the 1,3-bisphosphoglycerate and again at the phosphenol pyruvate step. Because this happens twice per glucose, glycolysis produces 4 ATPs. But since 2 were invested earlier, the net gain is 2 ATPs. So overall, glycolysis produces 2 ATP, 2 NADH, and 2 pyruvate. Pyruvate now enters the mitochondrial matrix. Here it undergoes oxidative decarboxylation by the pyruvate dehydrogenase complex. First, 1 carbon is removed as carbon dioxide. This is the first carbon being lost from the original glucose molecule. At the same time, NAD plus is reduced to NADH, again capturing high energy electrons. The remaining 2-carbon fragment is attached to coenzyme A, forming acetyl-CoA. This step is irreversible and highly regulated. Since 1 glucose produces 2 pyruvate, everything here happens twice. The Krebs cycle begins when acetyl-CoA combines with oxaloacetate to form citrate, a 6-carbon molecule. What follows is a series of oxidation reactions that gradually release energy. 2 carbons are removed as carbon dioxide, meaning by the end of this cycle, the carbons that entered as acetyl-CoA are completely oxidized. 3 NAD plus molecules are reduced to NADH. 1 FADH is reduced to FADH2. 1 ATP or GTP, depending on the tissue, is produced by substrate level phosphorylation. The key idea here is that most of the energy here are not released as ATP directly. Instead, it is stored as NADH and FADH2. Since 2 acetyl-CoA enter para-glucose, these numbers double. The electron transport chain is located in the inner mitochondrial membrane. NADH donates electrons to complex 1, while FADH2 donates to complex 2. As electrons are passed through the chain, energy is released. This energy is used to pump proton from the matrix to the intermembrane space. This creates an electrochemical gradient, also known as the proton motive force. Protons then flow back into the matrix through ATP synthase. The movement of protons drives the rotation of ATP synthase. which drives the formation of ADP from ATP and inorganic phosphate. Oxygen acts as the final electron acceptor at complex 4, combining electrons and protons to form water. Without oxygen, the whole chain would back up and stop ATP production. So respiration is essentially a highly controlled redox process. Glucose is gradually oxidized, electrons are transferred to carriers, and the energy released is used to generate ATP. Most ATP is not produced directly from glucose, but from the proton gradient generated by electron transport. Finally.